Good day to you from Inside Drug Discovery. This video describes the discovery program of MT7117, an agonist of the melanocortin 1 receptor. The discovery program is summarized based on a drug annotation article in the Journal of Medicinal Chemistry. The article was authored by scientists at Mitsubishi Tanabe Pharma in Japan. A link to the article can be found in the video description. Let's begin with the proposed drug target, melanocortin 1 receptor, MC1R. This receptor is a GPCR with 5 subtypes. MCRs play various roles. MC1R specifically is expressed in several cell types and involved in skin pigmentation, inflammation, and fibrosis. Indications of interest for the purposes of the drug annotation article include types of protoporphyria and systemic sclerosis, all conditions that can manifest as dermatological problems. MC1R's principal exogenous ligand is alpha MSH, alpha melanocyte stimulating hormone, which is a polypeptide. Many exogenous compounds targeting MC1R and other MCRs are also linear peptides or cyclic peptides. To date, these exogenous MC1R ligands have experienced issues of patient tolerability, and in cases of orally administered compounds, poor oral bioavailability. Consistent with these issues, the starting point for the discovery program of MT7117 was compound 5 from previous research at Mitsubishi Tanabe. Compound 5 has promising on-target activity, but poor membrane permeability as well as a safety liability with hERG channel inhibition. Since hERG inhibition is often associated with lipophilic amines and permeability can be reduced in charged structures, the researchers started by trying to reduce the basicity of the pyrrolidine ring on the left side of the compound. Reducing the basicity relied on exploiting a potential inductive effect by adding fluoro groups either on the pyrrolidine ring or its N-T-butyl group. Here are 3 compounds, 5, 6, and 7. As you can see, fluorination of the T-butyl group in compound 6 devastated potency, but note that membrane permeability rose dramatically. Ring fluorination in compound 7 improves potency somewhat, improved permeability over 10-fold, though still low, and slightly reduced hERG inhibition. But compound 7 had much poorer metabolic stability, which I don't show here, relative to compound 5. Compound 7, regardless, was pushed forward for further study. Compound 7 on the left has at least 2 issues. Membrane permeability, although improved relative to 5, remains low in a CaCO2 assay, and its metabolic stability is low. hERG inhibition is a concern as well. Replacement of the imidazole linker with the piperidine amide was hoped to improve permeability by reducing polarity. You can maybe envision the new amide on compound 7 by picturing a ring here and removing the rest of the imidazole ring. The outcome was compound 8A. Permeability was much improved. Surprisingly, metabolic stability also improved. The gains in these ADME properties help soften the impact of reduced potency. The researchers hope to claw back lost potency with optimization of other parts of the compound. Moving ahead with compound 8A, we can see a few changes to the aryl group on the pyrrolidine ring. Replacement of the fluorines with a paramethoxy restored the lost potency with minor changes to membrane permeability and metabolic stability. The researchers then turned their eyes to the N-T-butyl group on the pyrrolidine ring. Here are a few N-alkyl analogs. None of the new compounds could match the potency of the T-butyl group, but the cyclopentyl group in compound 9E was similar in terms of potency and a bit better for metabolic stability. Both of these R groups, T-butyl and cyclopentyl, were considered in the final round of optimizations. At the close of optimization, the researchers revisited the central piperidine ring. The best option was replacement of the 6-membered piperidine with a substituted 5-membered ring, pyrrolidine ring. As you can see, the researchers kept the door open for both N-T-butyl and N-cyclopentyl substitution. In the end, the N-cyclopentyl group gave the best compound, compound 10C, with preservation of membrane permeability being the deciding factor. Compound 10C was eventually formulated as its phosphoric acid salt with a research designation of MT7117. PK studies on MT7117 in male cynomolgus monkeys determined the compound's half-life, clearance, volume of distribution, and bioavailability. In addition to having favorable PK properties for achieving adequate oral exposure, MT7117 also showed promising selectivity for MC1R over other members of the MC1R family. Much of the in vitro and in vivo efficacy data of MT7117 has been previously reported in a separate publication, a link to which is at the bottom of the screen. Data from one study and the other article are summarized here. This 4-week treatment study followed how MT7117 induced pigmentation in the facial skin of cynomolgus monkeys. The values in the table were manually read from a graph and should be considered approximations. MT7117 shows a positive correlation between dose and the skin darkening response, reported as delta L star. Note that the highest dose group, 30 mg per kg, were only treated for 3 weeks. The pigmentation effects were reversible during a 16-week recovery period. Based on the favorable efficacy and safety data from this and other in vivo studies, MT7117 was advanced into clinical studies. MT7117 is currently being studied in clinical trials for treatment of 3 different conditions. The first 2 are EPP, erythropoietic protoporphyria, and XLP, X-linked protoporphyria. In EPP and XLP, patients experience pain when skin is exposed to sunlight. The third condition is systemic sclerosis, or scleroderma. This is a fibrotic disease in which one's skin hardens and tightens. At the time of publication, MT7117 has demonstrated proof of concept efficacy in phase 2 trials for EPP and XLP. MT7117 has been given a non-proprietary name of dersimelagon, which is formulated as its phosphoric acid salt. That is the story of the discovery program of MT7117, or dersimelagon phosphoric acid. MT7117 is a small molecule, orally available MC1R agonist for the treatment of certain skin conditions. Thank you for watching this video. If you have any comments or questions, please leave them below the video. Also, consider liking the video or subscribing to the channel. Take care.